Batrachocytrium dendropatitis, the frog chytrid, also known as BD. BD is a non-hostile parasitic chytrid fungus that has been associated with population declines of amphibians. BD has two life cycles. The first is dispersal as a motile waterborne zoospore. The secondary is a stationary monocentric thallus that develops into a single zoosporangia. Survival, growth, and reproduction is highly temperature dependent. Temperatures higher than 25 degrees Celsius increase the rate of epidermal turnover and reduce the growth of dendropatitis. BD is considered unusual because it's the only chytrid that parasitizes vertebrate animals. BD causes chytridiomycosis. Chytridiomycosis is a cutaneous mycosis that forms in wild and captive amphibians. It causes infections and cells within the outer skin layers that are keratinized. An amphibian infected with chytridiomycosis exhibits a wide variety of symptoms. The most common ones are reddened and discolored skin, excessive shedding, postures that keep the skin of the belly up from the ground, seizures, and nocturnal behavior.
One way the BD infection is transmitted is through zoospores. The zoospores require moist environments and can survive in these environments for several months. The zoospores can spread in water, moist soil, or on the skin of the infected organisms. The most common way the BD fungus spreads is from the direct contact of an infected amphibian with an uninfected amphibian. This occurs most often in territorial or breeding encounters. Humans wearing wet boots, vehicles, or livestock moving through infected sites and amphibian habitats can also cause BD to spread as they carry the fungus with them to uninfected areas. It is also found that in captivity, it is possible to house infected amphibians in enclosures next to uninfected amphibians and not transmit the infection as long as water and tools that are not shared between the enclosures.
BD infects amphibians through the inside cells of the outer skin layers, which contain large amounts of protein called keratin. Keratin makes the outside of the skin tough in order to prevent injury and is also what hair, feathers, and claws are made of. Chytridiomycosis makes the skin become very thick due to a microscopic change in the skin. Unlike most animals, amphibians drink water and an important electrolytes by absorbing them through their skin. Changing the ability of amphibian skin causes death. Without a sufficient amount of electrolytes in their bodies, the amphibians go into cardiac arrest and death follows. Some amphibians, like the lungless salamander, use their skin to breathe, so a BD infection will suffocate them. There are some amphibian species that are infected by BD but do not die. Species such as the African cloud frog and the American bullfrog manage to survive. A scientific reason for this has not yet been established, but it's possible that a special type of bacteria or poison in their skin prevents further development of the chytridiomycosis. The resistant species are key to finding a solution to this problem. However, these resistant species could also spread the fungus to other non-resistant species. A rough estimation of about 200 frog species infected by BD have been extinct in the past 30 years. Over 350 amphibian species are currently known to have been infected.
Since the discovery of BD, the chytrid has been found in virtually every continent sustaining an amphibian population. This includes South America, Central America, North America, Australia, Europe, Africa, and Asia. But the fungus is only actively spreading in Australia, Europe, South America, Central America, and Western North America. The chytrid is also absent in Madagascar, Borneo, and New Guinea. Due to its high sphere of influence on a global scale, the origins of BD have been a key point of research. With the current genetic and historical evidence, BD has been present in Africa, Western North America, and Japan for a long period of time, and have been proposed as possible sites of origins but the exact location is still unknown. This is the current figure of where BD is currently found on a global scale.
The high percentage of devastation of BD on the amphibian population has sparked the intensity of research regarding its control. Currently, the main focus of control is quarantine, avoidance of foreign water sources or infected amphibians. This includes limiting the foot traffic around sources of water known to have presence of BD or containing amphibians infected with BD. Another source of remediating includes sanitation of boots, tires, and equipment used while working near or in the hydrate to prevent the spread of BD. Aside from the conventional ways of control, there has been some research on BD-resistant amphibians. The American bullfrog and African clawed frog are both resistant to BD, but the source is still unknown. There is a multitude of hypotheses for the resistance, including the presence of some symbiotic bacteria discouraging the growth of BD, the production of antimicrobial peptides from poison glands, and genetic resistance. There has been a promising breakthrough with antifungal medication. The Smithsonian National Zoo has developed a series of baths in its drug itraconazole that mitigate the deaths of amphibians due to Batrachocytrium, but the large-scale use of itraconazole in the wild is not feasible.